So today we will start with the biology first chapter, or as such from the textbook the second chapter, that is structure of chromosomes, cell cycle and cell division. Now, so we had learned in the earlier chapter about the nucleus, that the nucleus of this particular cell contains the what we call it as chromatin fibers. So these chromatin fibers are the one which are going to be present in the cell when the cell is in an immature or a just born cell. So when the cell is in a phase which is called as a resting phase, at that time it is in a form of a chromatin fibers, which is just a network of fibers, network of threads. As it matures, the nucleus, the chromatin fibers over here, they are going to form what is called as chromosomes, which will be something like this. So this will be chromosomes which will be present in the cell or in the nucleus in this particular pattern. So there will be these chromosomes which are going to be there over here like this. So this I have shown is a cell nucleus having 4 chromosomes, 1, 2, 3, 4. Accordingly, each and every living organism has got a particular set of chromosomes. Like in case of human body, we have got 46 chromosomes. That means 23 pairs of chromosomes. This will be 2 pairs of chromosomes or 4 chromosomes. As I told you, we have got 46 chromosomes, that is 23 pairs of chromosome, out of which 23 you are receiving from your mother and 23 from your father. So when you talk about chromosome, it is seen that in interface, the nucleus shows a network of very long, extremely thin, dark, stained fibers called chromatin fibers. That's what is given in the textbook, that in the interface that as I told you is the resting phase of the cell, wherein the cell has just got duplicated and a fresh shell has been made. So that in that interface you can see it is just a dark, extremely thin fibers, dark staining fibers called as chromatin fibers. As the cell begins to enter the first stage, that is the prophase of the cell division, the chromatin fibers condense to form chromosomes. So these chromatin fibers condense, they wind up, they keep on winding up and they form into a condensed form which is called as the chromosome. So to form chromosomes, the chromosome readily pick up certain dyes and get colored, hence the name chromosomes. Chromo means colored and zone mean body. So colored body, that's why we call it as chromosomes, because they immediately take up the dyes, so it gets colored very easily. So chromosomes can be defined as highly condensed coiled chromatin fibers. So highly condensed coiled chromatin fibers are nothing but chromosomes. So when you talk about chromosomes, let's understand now the complete structure of the chromosome. So structure of the chromosome, it is this way, that each chromosome in its condensed form as visible during the start of the cell division consists of 2 chromatids, that is chromatid 1, this is chromatid 1 and this is chromatid 2. So these are 2 sister chromatids. So they are going to be forming into 2 sister chromatids connecting together to form 1 chromosome. So chromatids joined at some point along the length. This point of attachment, so this chromatid over here and this chromatid over here, they are going to be attached at this particular point. So this point over here is called as centromere. So centromere is the one which is connecting the 2 chromatids. So if I consider that this is one chromatid, this is another chromatid, if they connect it like this, and my knuckles over here, this point over here, this and this over here is the point which is called as the centromere. It appears as a small constricted region, that is over here, small constricted region, thoda narrow ho jata hai wahan pe, so that is constricted region. The centromere also serves to attach the spindle fibers during cell division. So when the cell division takes place, it is at this particular point that the spindle fibers are going to get attached to it. So these are spindle fibers. We'll understand what exactly the spindle fibers and all when we study about the cell division. But here we can understand one thing, that the centromere is the point where the two ropes or the two spindle fibers are going to get connected so that they can be divided into two parts. Each chromosome's centromere is located at a particular site. As the spindle fiber contracts, the sister chromatids are separated at each to pull away from to the other end towards the poles to divide the whole cell. So basically it is this way, that when the chromatin fibers are there, the chromatin fiber forms into chromosomes. At the centromere there is going to be the two spindle fibers coming over there, and these spindle fibers are going to contract, and as they contract they are going to pull apart the two chromatids. So if you disconnect it like this, then it is going to get apart to the two poles of the cell, forming the cell division. So and after the completion of the cell division, again this chromatin fiber, the chromosomes will unwind and become like chromatin fibers, because that is again the resting stage or the interface for the new cell which has got developed. Now talking about the chromatin, so chromatin material consists of two parts. One is the DNA and the second part is the histone. So the chromatin fiber or chromatin material, the chromatin thread, the thread of the chromatin. So each and every, the small thread what we are talking about, that thing is made up of two parts. One is the DNA and other is the histone. Now 40% of this chromatid, 40% of this is DNA and 60% is histones. DNA is deoxyribonucleic acid. Deoxyribonucleic acid. Okay, deoxyribo is one word, that is deoxyribo, and then it is nucleic acid. So N for nucleic acid. So this is nothing but DNA. So whereas the histones are particular kinds of proteins. Histones are balls of protein. They are protein balls. So these are the types of proteins. So these are proteins. Okay. So histones are proteins, whereas the DNA are the deoxyribonucleic acid. Now let's see in detail about the DNA and the histones and how the chromosome is being formed. So if you see, the chromosomes are such that, suppose I consider this as a chromosome. So this chromosome over here, actually each and every this part, chromatid over here, it is made up of thin lines over here. This is thin lines which are coiled and coiled and coiled, super coiled over here. So this thin line, what is there? This thin line is made up of the DNA and the histone. So DNA histone forms this particular thin line. And this DNA and histone is such a way that there are 8 histones. 1, 2, 3, 4, 5, 6, 7, 8. 8 histones which are bundled up. You know, they are going to be bundled up like this. They are going to be bundled up like this with the help of a ribbon-like structure that is nothing but the DNA. So DNA, ऐसा बोल सकते हैं कि ये 8 अगर यहाँ पे balls है, मैंने एक DNA का thread लिया और उसको ऐसे बांधा, ऐसे बांध रहा हूँ, I'm tightly getting, they are getting close to each other. So such a kind of bonding. So basically you can say that if I have made this, there are 8 histones over here, 1, 2, 3, 4, 5, 6, 7, 8, and I am just winding this up over here and forming this particular kind of a thread. So in this way you are forming a bundle of 8 balls tied up with the help of a ribbon-like structure called as a DNA. So this 8 histone and the DNA complete over here is forming the chromatin fibers. Now, so we can say that it is DNA is there. DNA लिया, 8 balls लिया, histones लिया, उनको tie किया. जैसे ही हमने उसको tie करते हैं, तो that particular 8 histones and the DNA forms the nucleosome. So 8 histones plus DNA, they form a nucleosome. Okay, there will be the formation of nucleosome with 8 histones and a thread like structure DNA binding that together. वैसे ही histones जब इसको coil करते रहते हैं तो it is going. So now over here we can see that a single human chromosome may have about 1,000,000 nucleosomes. So this nucleosome we will be having minimum a million of nucleosome in our this. So we can understand it this way, that a DNA strand winds around a core of 8 histone molecules. This core can be imagined like a football around which a long rope is wound with 1 or 2 loops. Okay, so if there are 8 footballs and they are winding it with 1 or 2 loops of the DNA, then each such complex thing over here is called as a nucleosome. So the entire chromatin fiber is coiled and supercoiled. Now then it is going to be coiled and supercoiled. You know, the telephone wire is coiled over there. So that particular coiled like structure, I think I got the thing. So yeah, you can consider it this way. This is coiled and supercoiled. So ये ऐसे बन रहा है, ऐसे था. और अभी उसको मैं coil करके पूरा का पूरा उसको इस तरह से बना दिया. So it is like this particular thing. So this is going to be like this as one chromosome. So this is one chromatid of the chromosome. ऐसे करके दो part बन के it may form into like things. Okay, so you can quite easily understand that how chromosome is being developed. So each entire chromatin fiber is coiled and supercoiled, something like a coil and supercoil as in a telephone cord, and we get this particular kind of a chromosome. So as I told you, we have got the 8 histones, DNA thread winding that 8 histones, forming a nucleosome. Such nucleosomes multiplying and forming into a thread-like structure, and that thread-like structure coiled and supercoiled to form a chromosome. So that was about the formation of chromosome. Now let's study how a DNA is formed, because we just said that DNA was a thread-like structure, or we can say DNA was a ribbon-like structure. So if you actually see a DNA, the DNA when you see it in a microscope, it is like 2 helical structures like this. So it is one thread-like structure like this and another thread-like structure like this. और ये each and every जो line दिख रहा है, that actually is made up of a double line over here like this and like this. So it is seen that it is a double stranded ribbons, one here and here. So that's why the thickness is there, and between this over here are going to be some nitrogenous proteins. So nitrogenous proteins will be there, like it will be G and then real C and then A and T and again G and C, so on. So it could be like this, that A, T, then again C, G, and again T, A, and so on. So, okay, so it can be seen that in this way the whole DNA is formed. So this particular thing is nothing but one DNA strand. So one DNA strand. So now let's understand what exactly is this DNA strand made up of. Now DNA strand यानी ये एक blue line and ये particular red line जो मैंने draw किया है, that is made up of, you can say that firstly there will be one phosphate. There is one phosphate molecule, okay, and this phosphate molecule is connected to a pentose sugar. This is pentose sugar. I think I better draw it here because I may need more space. So it is this way, that there is this phosphate which will be here, another phosphate over here coming from top, then there is pentose sugar which is like this, another pentose sugar over here, that is of the next strand. So this one strand, but this complete thing is one strand, so it is like this, connected like this, and then it will be connected further with another pentose here and another phosphate here and phosphate here. Now between this pentose sugar over here will be a base which is connected like this. So again you can see that there will be another pentose sugar over here connected to another base over here. So this is another base, this phosphate, and this is pentose sugar connected like this. So in this way you are forming into what we call it as the strands of the DNA. Now the strands of the DNA, so we can see that what are the content over here. One is the phosphate, second is the pentose sugar, and then over here are three things. So here we have got the third part over here, so this is part 3, which is the base. Now base is a nitrogenous base, so it is nitrogenous base. Now this nitrogenous base has got 4 kinds and they are connected to each other over here by a hydrogen bond. So this particular part is a hydrogen bond. This is a hydrogen bond. And over here can be 4 kinds of proteins. The 4 kinds of proteins are adenine, given as A. The next is guanine. This is G. The next is cytosine, which is C, and lastly is thymine, which is T. So the 4 nitrogenous bases, that is A, G, C and T, it will be seen over here that always the G combines with C, and when they combine with a triple bond sharing 3 electrons with each other. And when the other thing is A, it combines with T forming a double bond. It will never happen that the A combines with C or G combines with T. It will always be the G combining with C, that is guanine and cytosine combining with each other, and adenine and thymine combining with each other. Guanine and cytosine are going to combine with each other with 3 nitrogenous bonds, that is 3 triple bond, whereas over here adenine and thymine are going to combine with a double bond. So that's why we have over here. So as we go through this, what is the explanation given in the textbooks, let's go through one by one. First thing, the DNA is a very large single molecule. So it's a very large, this is one large single molecule. This is one single molecule. And hence it is described as a macromolecule, bigger, larger molecule, so macromolecule. It is composed of 2 complementary strands wound around each other in a double helix structure. So it is 2 complementary strands. So this is, as I told you over there, it was like this, that one strand like this and one strand like this. So there are 2 complementary strands which are going to be going in a double helical manner like this. So it is going to be going like this, ऐसे करके double helical structure में जा रहे हैं हम लोग. Each single DNA strand is composed of repeating nucleotides which are made up of 3 components. So this over here, this particular part, यहाँ से लेकर यहाँ तक, is called one nucleotide. So a nucleotide consists of a phosphate, pentose sugar and a nitrogenous base. Okay. So the nucleotide, which is made up of 3 components, phosphate, pentose sugar arranged lengthwise, and nitrogenous base attached to the sugar inwards. So attached to the sugar which is covered inwards. which extends to join by a hydrogen bond the complementary nitrogenous base from the other strand. So this nucleotide is going to combine with another nucleotide over here. So ये nucleotide को तो combine करने वाला part कौन सा है? ये particular nitrogenous base का hydrogen bond. So the hydrogen bond over here is going to act as a combination between the two. So yeah, I just made one thing, and that is this line over here should be going like this. So this is one nucleotide. Pentose sugar, phosphate and nitrogenous base over here. ये तीन मिलके एक न्यूक्लियोटाइड बनेगा. ये दूसरा न्यूक्लियोटाइड बना. ये दोनों कंबाइन हुए यहाँ पे with the help of a hydrogen bond. Thus the two strands together make a ladder-like arrangement with the nitrogenous bases forming the rungs of the ladder. So you can see if this is the ladder-like structure, these are the rungs of the ladder. Okay, you can step on it. So that is the rungs of the ladder. The bases adenine, guanine, cytosine and thymine. Adenine pairs with thymine with a 2 hydrogen bond. Adenine with thymine with 2 hydrogen bond. Whereas the guanine pairs with cytosine with 3 hydrogen bonds. Okay. So the formation of a new DNA will be there. That there will be during the interphase, the DNA molecule is going to split and generate into new ones. So it is just going to be this way. यहाँ पे ये जो है वो आगे आके split हो जाएगा, ये आगे जाके split हो जाएगा, and it will be formation of new ones over here and new ones over here, which is not in syllabus as such, so not to worry about. But there is a replication. They are given you in the figure 2.7 how the DNA is going to replicate. So the new forms, the replication is going to take. यहाँ से ये हो रहा है, तो ये यहाँ जाके दूसरा दो बनेगा और यहाँ पे ये दूसरा दो बनेगा. ऐसे करके दो यहाँ पे अलग-अलग DNA form होंगे. So this is how the new DNA strands are going to be formed over here. So that's about the structure of the DNA. We can just go through very quickly about this thing. अगर strand से चालू किया तो it is this particular one nucleotide consists of phosphate, pentose sugar and nitrogenous base. Another phosphate, pentose sugar and nitrogenous base combining with the hydrogen bond. This particular thing is going to be forming the DNA strands, double helical structure. 8 histones रहेंगे, proteins. They are going to be tied up with this DNA strand and forming one nucleosome. These nucleosomes are going to multiply and are going to be coiled and supercoiled and all, and to form into a chromatid, which on supercoiling becomes a chromosome. So this is how the chromosome or structure of the chromosome is going to take place in any living organism.